Hello everyone, my name is HENG LIM and I'm currently studying biotechnology at Taylor's University. When I think about biotechnology, I think about its multifunctionality and how it can be utilized for various purposes. I think biotechnology is the future and our way forward, especially when we look at its ability to aid in ecology and conservation efforts. Molecular mapping or genome mapping is often used to identify and analyze the genetic maps of organisms using several biotechnology techniques such as locating quantitative trait locus or QTLs using molecular markers and also analyzing restriction fragment length polymorphism by cloning DNA sequences with radioactive probes. Molecular mapping and DNA marker technology is very promising when it comes to wildlife conservation, as these techniques can accurately identify and distinguish different species to allow scientists to properly plan and execute conservation efforts. In the past, species identification and classification, such as Aristotle's system and the Linnaean system, were based on morphology, or physical traits that can be seen by the naked eye, such as the color of blood, the number of legs, or the presence of scales. As such, this method of classification proved to be a limitation as several species undergo drastic morphological changes, such as the caterpillar, which transforms into a butterfly, boasting a completely different physical appearance. Besides that, the existence of cryptic species also makes species identification and classification even more challenging, as two very different species could closely resemble one another, looking so identical that even expert taxonomies cannot tell the difference by simply looking at it. Due to the limitations presented by the morphological classification of species, a more reliable way to establish the different species is by looking at the molecular aspect and distinguishing each species by their genetic compositions. As such, molecular mapping plays such an important role in species identification, which ultimately helps in improving conservation efforts, as scientists are able to resolve taxonomic uncertainties when creating species management plans to ensure that in situ or ex situ conservation methods directly address the affected species. This is because each species has unique and distinct requirements such as its habitat, diet, temperature, and even humidity, in which misidentification of species can be extremely detrimental to its survival. On that note, molecular mapping is essential in analyzing the effects of environmental stress and also the adaptations of species in order to overcome these stresses, which provides pivotal information for wildlife conservation. For instance, molecular biomarkers are used for ecotoxicological analysis of wildlife like rock oysters, moon jellyfish, and even various plants to study the reactions of these species to various abiotic stresses such as chemical contamination and even poor water quality. As such, scientists can also exploit these biological processes to understand the extent of damage to wildlife while also investigating the adaptations, such as behavioral changes to avoid environmental stress and alteration to their reproductive behavior. This information then allows scientists to study the evolutionary history of various species, which positively impacts conservation efforts. Apart from that, understanding a species from its molecular components to physical requirements can greatly help conservationists, as it not only allows for better management plans, but also provides accurate information to implement anti-poaching laws towards protected and endangered wildlife. In this case, biotechnology can be utilized to identify illegal poachers for prosecution under illegal poaching laws, as trace evidence of wildlife DNA can be extracted from the field or even on the belongings of the poachers, and then identified using molecular mapping techniques. Traces of DNA from threatened and endangered wildlife presents crucial evidence to charge illegal poachers, which is a big step for conservation efforts, as it was previously very difficult without strong evidence tying these criminals to the crime. As of 2020, there are over 35,000 threatened species of wildlife on the International Union for Conservation and Nature (IUCN) Red List, as opposed to about 30,000 species in 2019, highlighting the increase of over 5,000 threatened species and also extinction of approximately 24 species in just 1 year. This illuminates the need to further improve our pre-existing conservation techniques by incorporating a variety of biotechnology techniques to better manage as well as prevent species extinction. It is essential to conserve wildlife and maintain their genetic diversity, and I believe that biotechnology has a great potential in ensuring the survival of threatened and endangered species. As such, I believe that biotechnology is the future and the way to move forward. Thank you.